So good morning, dear aspirants of the NET. In the previous section, we have discussed the overview of the cell signaling, its importance and the major players. We have also discussed the entire signaling cascade, right from the ligand to the receptors, signal transducers, effectors and secondary messenger. In this particular section, we will discuss the entire relay of the signaling molecules from secondary messenger to the entire effect that is being brought about. Additionally, we will see the importance of the receptor tyrosine kinase, which is one of the very important category of receptors. Particularly, they are involved in cell growth and division and differentiation. And we will also see a particular kind of signaling molecule, that is nitric oxide or NO. Since it is recently in last decade, the discovery of NO as a signaling molecule has resulted into a very enthusiasm in the exploration of nitric oxide in the signaling cascades. So that might be one of the reason that this particular section, that is nitric oxide, is also being asked in NET exams very frequently. So we will see this entire things in this particular section. So as far as transmitting the signal is concerned, protein kinases plays a very important role. Here we have to keep in mind that kinases, wherever the term kinase would come, that means phosphorylation. Kinase are the category of enzymes which will add a phosphate group to its substrate. There are variety of protein kinases, that might be soluble ones, there might be receptor or transmembrane kinases. In this particular cases, they will be all soluble protein kinases. There are variety of protein kinases doing variety of cellular activities, but their job is specific, that is the phosphorylation of the substrate. We have already discussed that in the cellular systems, phosphorylation plays a very important role. Phosphorylation will take place either at tyrosine residue or serine or threonine. Now, only these three amino acids can be phosphorylated. So very obviously, only these three can be dephosphorylated. Now this phosphorylation and dephosphorylation are more or less like a molecular switch. In some of the enzymes, moment you add a phosphate group, obviously it will be added by a kinase. Moment phosphate group is added to that particular protein or that particular enzyme, it will become active. And now to inactivate it, you have to just hydrolyze that particular phosphate group, or you have to remove that phosphate group. So moment that phosphate group is cleaved or detached, it will be inactive. That will be brought about by phosphatase. Now, it is not a universal thing in the sense that it's not always like phosphorylation will activate and dephosphorylation will inactivate. Actually, vice versa is also true. In some of the enzymes, what happens is exactly opposite. Moment they are being phosphorylated, they will become inactive. And moment they are being dephosphorylated, they become active. So, if you will phosphorylate it, they will become inactive. You will dephosphorylate, they will become active. So, as we have seen in the home, if there is switch, if we push it downwards, it is switched on. If we push it upwards, it is switched off. But in the power switches, it is exactly opposite. When we push it down, it is switched off. When we push it up, it switches on. So, same way, these molecular switches are being there in the cell, and it is phosphorylation and dephosphorylation event. Now, the phosphorylation event is brought about by the protein kinases. It is a very important category of enzyme, and that is why it is being exploited in the signaling cascades to bring about the activation of variety of enzymes and proteins. If we see any of the pathway or the metabolic pathway, what we will see is that there is one regulatory enzyme. That particular regulatory enzyme would be activated or inactivated by kinase or phosphatase, that you can see across the spectrum. and this particular activity will be brought about by kinases. So the phosphorylation frequently serves to activate the substrate of the kinase, and it will also target the substrate for the degradation, thereby inactivation. So there are variety of phosphorylation cascades. In this particular diagram, we can see a phosphorylation kinase cascade where one particular kinase is activating another kinase, in turn activating another kinase. So kinase 1 will activate its substrate, that is kinase 2, by phosphorylation. Moment it is being phosphorylated, it will act the third one, which is against a kind of kinase, and it becomes active on phosphorylation and it will now work on its substrate. So such kind of phosphorylation cascades, one of the very important phosphorylation cascade is MAPK, that is mitogen activated protein kinase cascade. This is mostly involved in cellular division and growth. So such kind of cascades are also involved in some cases. It's not like that a protein kinase and directly a particular enzyme. It might be protein kinase, another protein kinase, another protein kinase, and then its substrate. So such kind of cascades are also being seen in the signaling cascade. Now this particular question is based on the fact that whatever signaling cascades which are being governed by visual stimuli, that is through eyes or images, they are being governed by a particular category of receptors, which is GPCR. Now in this particular question, the question is indirectly being asked on GPCR only. We can see rhodopsin is a transmembrane protein belonging to the large family of G protein coupled receptors. It is found in the disc of rod cells of human retina. Activation of rhodopsin is due to how these particular rhodopsins are being activated. The option A, phosphorylation of its extracellular tyrosine residue. Option B, binding of the external ligand to its extracellular loop. Option C is photoisomerization of the prosthetic group, and option 4, binding of the calcium ions to the transmembrane aspartic groups. Actually, when we will see the entire pathway where rhodopsin is involved, it is the photoisomerization of the prosthetic group, because it is the light which will serve as a ligand. In this particular table, which is taken from molecular biology of the cell, we can see variety of GPCRs and their effectors. The notable thing about the GPCR is that as far as their G protein is concerned, if that particular alpha subunit is GS alpha, that means it is stimulatory alpha. That means it will result into the activation of the effector and cyclic AMP level will rise. If it is inhibitory alpha, GI alpha, as in the case of certain alpha 1 adrenergic receptors, what you will see, the activation of those G protein will result into the decrease in the concentration of the cyclic AMP. That is, they will inhibit the effector molecules. Similarly, G alpha, G OLF, G olfactory receptors are there, which will bring about the olfactory signals molecules. In this particular diagram, we can see the stimulation and inhibition of the G proteins. In one case, it is being stimulated because the G alpha unit is GS alpha, and on the right hand side you can see it is GI alpha. That is why there is inhibition of the particular effector molecule. So this particular adenylyl cyclase is an effector, but in one case by one receptor, which is GPCR only, its activity is increased. In another case, where again it is a GPCR, but its activity is reduced. So in the biological systems, no doubt there are variety of G proteins, there are variety of GPCRs, and G proteins remain same, but this G protein will employ variety of G alpha subunits, and by that the same G protein having different kind of subunits of G alpha or its subtypes, it can carry out cell signaling pathway for variety of receptors. In some cases, it may involve adenylyl cyclase. In some cases, it may involve guanylyl cyclase. In some cases, it may involve phospholipase C. So it is the same GPCR, but it can employ variety of effectors, thereby variety of secondary messengers in variety of cases. Now in this particular question, it is being asked that in the cyclic AMP pathway, G protein will stimulate what? Phospholipase C, adenylate or adenylyl cyclase you can say, the endoplasmic reticulum or calmodulin. Obviously it is adenylyl cyclase, and we have already seen in the previous slide that G protein will stimulate the adenylyl cyclase, and thereby cyclic AMP levels would be, in some cases it might increase, in some cases it might also decrease. There are variety of ligands for the GPCRs. You can see variety of neurotransmitters, variety of hormones can serve as a ligand for the GPCR. But in variety of cases, it will employ variety of G proteins or their subunits to bring about the required change in a particular case. So it will not be like that the effect is required for epinephrine and some other effect is being brought about. It will never happen, because a particular kind of ligand will have a particular kind of affinity for GPCR and particular kind of G alpha subunit. Now, one interesting thing is that when G alpha comes and bind to the adenylyl cyclase, what G beta and G gamma would be doing? In some cases, GPCR can also serve as ion channel, where the activation of the ion channel would be brought about the other two subunits, that is G beta and G gamma. So they can also serve as ion channels, or they can activate certain ion channels. Now in this particular question, it is being asked all of the following statement about the monomeric G proteins are true, except. That means we have to find out the statement which is incorrect. First, option A, they are regulated by GDP exchange protein. It seems to be correct. They are regulated by GTPase activating protein. We will see in the coming section that it is also correct, because they are also being regulated by certain proteins which are known as GAPs or GTPase activating proteins. Third, they regulate the vesicle fusion. And fourth, they help in uncoating of proteins from the vesicles. So option D is incorrect, or it is not true. Rest of the 3A, B and C are correct. This is the diagram for receptor desensitization. This receptor desensitization is a very important concept. Sometimes, even in the presence of the ligand, receptor would not transmit the signal. That means it is dishonoring the signal. Actually, as we have already seen, that there is a phenomena of crosstalk. That is, one particular signaling cascade will involve variety of molecules. Now, suppose in a particular pathway, one negative signal has come. Suppose a cell needs to divide, but mitochondria has not been duplicated. In that case, a message will be given to the interior of the cell that it is not the right time to divide. Similarly, if some mutation or genetic damage has taken place, it might be not the right time to divide. In such cases, the entire cascade of the signaling for the cell to divide would be brought to a halt by a process that is called as receptor desensitization. That means receptor is not honoring the presence of the ligand. Even if the ligand is present, receptor will not do the needful. Why? Because of the good of the cell. Because there is one or other reason for that particular pathway to stop. Now, how it will be brought about? How a receptor will become desensitized? It's all chemical bonds. It is all chemical modifications. So, receptor has to honor the ligand. But what happens? There are certain phenomena. Either there are certain molecules like arrestins that will bind to the activated receptor and then it will inactivate. So that is called as arrestin. In some cases, it can be sequestering. Sequestering is a kind of hiding. So might be the receptor will be, in a hideout, it will be transferred to a particular vesicle. So even though it is present in the cell, but it is not present for any function. In some cases, a receptor may also be degraded by one or the other protein molecule so that it will not bring about the cell signaling cascade. So, for the good of the cell, in some cases, a receptor may also be desensitized either by sequestering or by degradation or by inactivation by one or the other proteins. So, this is also a very important phenomena in the signaling cascades that is known as desensitization. Whenever desensitization is there, there might be one or the other valid reason for a receptor to be desensitized. In this particular question, it is being asked which of the following can serve as secondary messenger. Actually, you can see all these are ions. So, interestingly, one of the ion, that is calcium ion, it can serve as secondary messenger. So, among sodium, calcium, magnesium and copper ions, actually calcium ions can serve as a secondary messenger and it will mediate its signaling cascade via another protein that is called as calmodulin. We will see that particular pathway. In the eukaryotes, which of the following is not a secondary messenger? 1,2-diacyl glycerol, also known as DAG, is a secondary messenger. Cyclic AMP is also a secondary messenger. Inositol 1,4,5 triphosphate, it is also a secondary messenger. Now the option D, that is cyclin. Here actually correct answer is D, cyclin. It's not C. It is D, cyclins. It is not a secondary messenger because it is a protein which is involved in the division of the cell. It is the same question with the correct option, that is D. Which of the following compound does not act as secondary messenger during signaling processes? So here among cyclic AMP, calcium ions, inositol 1,4,5 triphosphate, it is triacyl glycerol which is not the secondary messenger. So option D is the correct answer. Besides that, cyclic AMP, calcium ions and IP3 whose full form is inositol 1,4,5 triphosphate, all these three are secondary messengers, but triacyl glycerol is not a secondary messenger. In fact, diacyl glycerol, DAG, is a secondary messenger. So here the correct answer is D. Now, as far as the features of the secondary messengers are concerned, they are usually small water soluble compounds which are, in some cases diacyl glycerol is an exception. It is not water soluble, it is lipid soluble, that will execute the message within the cell. These are also termed as secondary messengers because the primary messenger is the ligand molecule that remains outside the cell. Within the cell, an important entity that carries out the entire signaling is secondary messenger. So these secondary messengers plays a very important role in signaling cascades, and by acting on variety of protein kinases, they will bring about the required effect. Now, what are the four classes of the secondary messengers? Within the cell, the first major category is cyclic nucleotides. For example, cyclic AMP, cyclic GMP, they serve as secondary messengers. Whenever the effector is adenyl cyclase, the secondary messenger would be cyclic AMP. Whenever the effector is guanylyl cyclase, the secondary messenger would be cyclic GMP. So cyclic AMP and cyclic GMP, they are cyclic nucleotides which serves as secondary messengers. Similarly, there are certain lipid derivatives like diacyl glycerol or DAG and inositol 1,4,5 triphosphate, that is IP3. These are being generated when the effector molecule is phospholipase C. Whenever the effector is phospholipase C, it will result into generation of two secondary messengers, diacylglycerol and IP3. Among these two, diacylglycerol is a membrane-bound secondary messenger. This particular fact is also being asked in certain examinations. Now, one of the important secondary messenger which is an ion is calcium ion. So, calcium ion can also serve as secondary messengers. Similarly, a gas or gaseous molecule which can serve as a secondary messenger is nitric oxide. This is not an all exclusive list, but it is a representative among gaseous, ions, lipid derivatives and cyclic nucleotides. So mostly these are the secondary messengers which will bring about the effect within the cell. So you will see there are certain questions that are being asked on these particular aspects. Now, which of these secondary messenger is membrane bound? So cyclic AMP is soluble, IP3 is soluble and cyclic GMP is also soluble. Only diacylglycerol is a particular secondary messenger which is bound to the membrane. So the correct answer is C, that is diacylglycerol. Now, cyclic AMP as a secondary messenger, whenever adenylyl cyclase would be effector, it will use ATP as a substrate and would result into generation of cyclic AMP. Cyclic AMP is a secondary messenger and its rise in the concentration will bring about a particular change according to its concentration. If there is high concentration, the effect will be brought about very fast. If there is low concentration, effect will be brought about very slow. Now again, here one of the important thing is that there is an enzyme that is known as phosphodiesterase or cyclic AMP phosphodiesterase. It will convert cyclic AMP into AMP. So even if cyclic AMP is there, the signaling cascade can be stopped by the employment of phosphodiesterases, which will convert cyclic AMP into AMP. AMP is not a secondary messenger. Cyclic AMP is. So if you convert cyclic AMP to AMP, you can convert a secondary messenger into just a molecule. So if you want to stop a signaling cascade even after the generation of secondary messenger, we should convert it into some another form. That is possible in the case of cyclic AMP by the employment of phosphodiesterase, and that is why there are certain questions which are being asked on the phosphodiesterase. Here in this particular part, we will see the various features of the secondary messengers. Secondary messengers, they are very short-lived. They remain active for some microseconds, and then they bring about the change whatever is required. They will activate certain protein kinase and then they will become inactive. So if we need to activate so many protein kinases, there should be a transient increase in the concentration of the cyclic AMP, then only the continuous effect would be brought about. There is a rapid alteration in the concentration that will bring about the strong effect, either increase of a particular activity or decrease in a particular activity is governed by its concentration, and its concentration is altered very rapidly. The removal or degradation of the secondary messenger will terminate the cellular response in most of the cases, and message is mostly governed or encoded by its concentration. Whenever the cyclic AMP is the secondary messenger, obviously the effector would be this adenylyl cyclase, which is a membrane bound enzyme, will use ATP as a substrate and would generate cyclic AMP as a product, and this cyclic AMP will serve as a secondary messenger. Depending on the turnover number of the adenylyl cyclase, there will be increment in the number of secondary messengers, and the entire pathway would also be amplified manifold. Now this one particular thing that is being asked, CRAB. The full form of CRAB is cyclic AMP response element binding proteins. So CRAB is an ion chelating protein, ion scavenging protein, DNA binding protein or creatine binding proteins. So it is option C which is correct, a DNA binding protein. Actually once cyclic AMP is activated, it will activate protein kinase A, and this protein kinase A might activate a particular category of proteins which is DNA binding protein or transcription factors, that is known as CRAB, that is cyclic AMP response element binding protein. This will bind to the DNA and the modulation of that particular gene would be brought about. So in this particular case, CRAB is a transcription factor, and obviously a transcription factor has to be a DNA binding protein. So option DNA binding protein is correct. Now in the next question, we have to choose the incorrect statement. Means out of these four options, one particular statement is wrong. Phospholipase C generates DAG and IP3 from phosphatidyl inositol 4,5 bisphosphate. It is correct. Phospholipase C is not a membrane bound enzyme. It is incorrect. Diacyl activate DAG activates protein kinase C under increased calcium ion levels. It is also correct. IP3 induces the opening of calcium ion channels present on the endoplasmic reticulum, that we will see in the coming section. It is also correct. So here the incorrect statement or the correct answer is B. Phospholipase C is not a membrane bound. Actually it is a membrane bound receptor. Now diacyl glycerol activates what? Protein kinase A, protein kinase C, MAP kinase or tyrosine kinase. The correct answer is B, that is protein kinase C. We have to keep this thing in mind that protein kinase A is activated by cyclic AMP. On the other hand, protein kinase C is being activated by DAG. Similarly, in this particular question, which one of the following is involved in cell signaling process? Cytochrome P450 or protein kinase C or zinc finger or all of these. Actually it is protein kinase C which is involved in the signaling process. Cytochrome and zinc fingers are not involved in any of the cell signaling processes. So the right answer is B. Likewise, we have already discussed this particular fact. Calmodulin activates protein kinase in response to transient increase in cyclic AMP, calcium ions, diacyl glycerol or NO. The right answer is calcium ions. Actually, you have to keep this thing in mind. Whenever calcium is involved as a secondary messenger or as a signaling molecule, it will involve calmodulin. So calcium calmodulin complex will then relay or initiate a particular signaling cascade. So the correct answer is B. Now in this particular questions, again something is being asked about calcium ions. Which of the following statement concerning calcium ion is correct? Intracellular concentration of calcium ion is higher than that of extracellular fluid? No. The concentration of cytosolic calcium ion is transiently decreased by the activation of phospholipase C. Actually it is not correct, because there will be an effect on PKC. The effect of calcium ion is most often mediated by calmodulin. It is the right option, and we have also seen a similar question in the previous slide. And the fourth one, intracellular calcium ion is mostly free. Actually it is not like that, because whenever there is an increase in the calcium ion concentration in the cytosol, it will be sent to endoplasmic reticulum. So there will be increase in the concentration of calcium ions in the lumen of the endoplasmic reticulum, and the endoplasmic reticulums are being governed by certain receptors which are called as rhinodine receptors. These rhinodine receptors become accessible either via IP3 or calcium ions. So once calcium ions or IP3 bound to it, they will open and allow the calcium ions from the lumen of the endoplasmic reticulum to the cytosol. So sometimes it is also being called as CICR, that is calcium induced calcium release. That is the release of calcium induced by calcium ion itself. So the effect of calcium ions are most often mediated by calmodulin. This is correct. With respect to the plasma membrane, most enzymatic receptors are single pass, they are present entirely on the surface, they are 7-pass or they are multi-pass. Mostly the receptors which are doing dual purpose, they are serving as extracellular domain on the cell surface and they are having enzymatic activity on the cytosolic side, they are mostly single pass. Whereas other kind of receptors, which might be ion gated channels or GPCRs or serpentine receptors, they will be passing through 2, 3 or 7 times like that. So the right answer is A, or the single pass. Now in this particular thing, receptor dimerization and autotransphosphorylation. This is an important aspect of a particular category of receptor which is called as receptor tyrosine kinase. We already know the function of kinase. The function of the kinase is to phosphorylate its substrate, thereby activating or inactivating it. Tyrosine means when the tyrosine residue is being phosphorylated, it will be called as tyrosine kinase, and then it is serving as a receptor. If the tyrosine kinase activity is inbuilt with a particular receptor, then it is called as receptor tyrosine kinase. Receptor tyrosine kinase is one of the another important category of receptors which is being employed in the eukaryotic systems besides the GPCRs. Whenever we talk about cell growth, cell division or cellular differentiation, mostly receptor tyrosine kinases are involved, and these receptor tyrosine kinases would be scattered on the cell surface. Moment their ligand comes, they will dimerize, and once they dimerize, what happens? The one particular unit will stimulate the phosphorylation activity in the another unit, and another unit will bring on the respective unit. So the two units of the receptors, they will phosphorylate each other, and that is called as autotransphosphorylation. Means auto means from self, trans means of each other, and phosphorylation is the addition of the phosphate group. So this is very remarkable feature of RTKs, and this particular fact is asked in so many examinations. Which particular receptor is capable of autotransphosphorylation, or in some cases they just ask transphosphorylation. So we have to keep in mind that RTK is a kind of receptor which is capable of autotransphosphorylation, but this autotransphosphorylation would be bring about only when receptor will dimerize, and receptor will dimerize only when the ligand is present. So in the presence of a ligand or the signaling molecule, RTK will dimerize and then it will autotransphosphorylate each other. This is a typical example diagram of a particular signaling molecule that is being brought about by RTK. You can see these two green receptors, they dimerizes and then they transphosphorylate each other. Then they will activate a particular kind of signaling molecules which are called as RAS and RAF. These RAS-RAF will then activate MAPK or mitogen activated protein kinases, and we have already seen in the earlier section of this talk that MAPK will result into a phosphorylation cascade where one kinase will activate another kinase, will activate another kinase, and then after 3, 4, 5 times such kind of things are being repeated, and then the final substrate is being activated. Now we will see one particular question on receptor tyrosine kinase. So it is asking about the features of receptor tyrosine kinase. So the question is, receptor tyrosine kinase, option P, have a cytoplasmic kinase domain, very correct, because it is having the enzymatic activity. Dimerizes upon ligand binding, very correct, that we call as dimerization. Change conformation of their cytoplasmic domain upon ligand binding, very true. And then finally become catalytically active after the conformational change, very true. They will bring about the autotransphosphorylation. So all these 4 statements about receptor tyrosine kinase are true or correct. So the correct option is D. P, Q, R and S all are correct. Now we have to find the correct statement of these. The first one is RAS is a monomer GTP binding switch protein. It is correct. RAS is involved after this particular autotransphosphorylation takes place. Then phosphodiesterase converts cyclic GMP into GMP. Yes, it is very correct, because phosphodiesterase will convert any such kind of cyclic nucleotides into normal one or non-cyclic ones. Third one, receptor tyrosine kinase dimerizes after binding with the ligand. We have already seen that it is correct. And then receptor tyrosine kinase after activation stimulates RAS MAP kinase pathway. Yes, via RAS RAF pathway it will activate MAP kinase pathway. So option D, that is all these statements are correct. Now in this particular section a question is being asked, most common feature of RTK family of receptor is, we have already discussed that. Ligand induced dispersion? No. Ligand induced dimerization? Yes. Ligand induced trimerization? No. Ligand induced downregulation? No. So actually the right answer is option B, that is ligand induced dimerization, and this dimerization would then result into autotransphosphorylation. So this is a very important feature of the RTK family of receptors. Now this is the entire protein kinase summary. Actually there are more than 200 different kind of protein kinases in the cell. They are doing one or the other job. They are involved in variety of functions. This is evident from this particular diagram. Now in this particular question, nitric oxide, there is a question on nitric oxide. Nitric oxide would activate protein kinase G, protein kinase C, protein kinase A and protein kinase NO. Here protein kinase G is a correct answer. Whenever nitric oxide is serving as a signaling molecule, it will be relaying its entire process via protein kinase G. Here it is worth remembering that whenever calcium ion is the signal molecule, it will work via protein kinase C. Whenever there is a cyclic AMP as a signaling molecule, it will work via protein kinase A. Whenever there is nitric oxide, it will work or it will bring about the effect via protein kinase G. So this again tells us that in variety of signaling molecules, variety of protein kinases are employed so that required effect depending on a particular case can be brought about. Here it is also worth mentioning that this nitric oxide is an intracellular signaling molecule that is being asked in some cases. We can see in some questions. Which of the following is a component in the signaling pathway stimulated by receptor tyrosine kinase? Adenylyl cyclase? No. It is involved in GPC and other pathways. Adapter proteins? It is also not correct. Autophosphorylating receptor? They will come earlier in the earlier part. RAS activating proteins? So that is correct. The RAS activating proteins would be stimulated by receptor tyrosine kinases. Now in this particular part, it is being asked, IP3 gated calcium ion channels are present on. We have already discussed that whenever we have extra calcium in the cytoplasm or the cytosol, the extra or higher concentration of calcium ions in the cytosol is very much dangerous and it is considered to be toxic. So whenever there is extra calcium present in the cytosol, there are machineries that will pump these calcium ions in the lumen of the endoplasmic reticulum. So in a particular manner, they will be sequestered from the cytosol into the vesicles or the endoplasmic reticulum. So and then they are being gated by IP3 channels. So IP3 receptors will bind to inositol triphosphate or IP3, or in some cases certain special kind of receptors called ryanodine receptors will bind to calciums. They will open up whenever the calcium is required for some signaling pathway or some other thing within the cytosol. These channels will open up and from the endoplasmic reticulum calcium will come into the cytosol. Whenever there is high increase of the calcium ion concentration that reaches a toxic level, the calcium will be pumped back into the lumen of the reticulum. So the right option here is endoplasmic reticulum, which serves as a storehouse of the calcium ions. This particular diagram, there are questions were being asked on calcium and calcium related issues, can be understood from this particular diagram. Here we can see a particular receptor, that is phospholipase C, on activation will result into activation of IP3. These IP3 can bind to these receptors, which are called as IP3 gated calcium ion channels. Sometimes they are also called as ryanodine receptors. And then when this binds to these receptors, it results into a phenomena that is called as release of calcium ions by IP3 gated ion channels. And these calcium ions will now come into the cytosol. Now they will activate protein kinase C by binding to calmodulin. Obviously we know that whenever calcium ions are there, they will work via calmodulin, resulting into the activation of protein kinase C, and these protein kinase C will result into activation of certain enzymes or certain proteins. So this particular calcium ion will be stored in the endoplasmic reticulum. They are being gated by certain receptors. These receptors will open up when IP3 will bind to them, and then calcium ion will come into the picture and they will regulate the entire signaling cascade as the case may be. Now there is a question of MAP kinase. We have to understand one particular aspect of MAP kinase, that whenever we are talking about division, there will be involvement of MAP kinase. Mitogen activated protein kinases, and mitogen are those molecules or substances which will result into, or they will bring about the division of the target cell. So if you will put a cell along with mitogen, it will start dividing. So they will bring about the division by a cascade which will involve MAP kinase. So here it is asking about the features of MAP kinase. More than one option again can be correct here. Are activated by RAS pathway. Next one, phospholipase their targets on tyrosine residue. Act to stimulate or repress the transcription of target genes through the phosphorylation of transcription factors. It appears to be correct. And then become active when phosphorylated by MEK kinase. So option P, R and S is correct, and this particular one, Q, phospholipase their targets on tyrosine residue, is not correct. Now the receptor for nitric oxide is intercellular, intracellular, extracellular or transcellular. So the receptor for nitric oxide is intracellular. In some cases they may also be considered as transcellular, but looking into the entire aspect of the signaling cascade which is being brought about by nitric oxide, the more appropriate answer would be intracellular. Which of the following is not true about NO or the nitric oxide? First thing, it acts as intracellular signaling molecule as well as neurotransmitter, so it is very correct. Secondly, it regulates the vasodilation, it's correct. Synthesized from L-arginine, it's correct, it is being synthesized from L-arginine. The fourth, it induces adenyl cyclase which catalyzes cyclic AMP formation. This is incorrect because it does not involve that particular pathway. It will work via protein kinases C. That particular pathway would be brought about. So option D is incorrect, and in this particular case is the right answer, that it is not true about NO. Similarly, in this particular question it is being asked, which of the following statement is false for the nitric oxide gas? It is an intracellular signaling molecule. Deamination of histidine result into nitric oxide production. It's not like that. Actually, L-arginine is involved. So here stimulates guanylyl cyclase to produce cyclic GMP. It is also correct. Can be produced by activated neutrophils. It is also correct because there are nitric oxide synthetase enzyme present in the neutrophils, because they will be involved in the generation of reactive oxygen and nitrogen radicals. So there also they are involved. So deamination of histidine resulting into the nitric oxide production, this option appears to be incorrect or false. So this was all about the cell signaling process. We have discussed variety of receptors right from GPCRs, RTKs and JAK-STAT. Now in a nutshell, we are summarizing the entire talk. The first thing is that cell signaling plays a very vital role in the signaling in the entire cellular processes. Any aspect of cell's life, whether it is division, whether it is death, whether it is replication of the genome, whether it is duplication of the organelles, is all being governed by cell signaling cascades. A cell will communicate with another cell, which might be in the near proximity or it might be situated far apart, via cell signaling process only. The coordination and regulation in the case of multicellular organism is brought about by cell signaling pathway only. Not only a cell will communicate processes, the initial small signal will be amplified manifold to bring about a strong or robust response by the cell signaling pathway. Plus, cell signaling will activate so many proteins or so many enzymes, or it will diversify the cascades so that the effect will be brought about very strongly and rapidly. Because of all these things, cell signaling plays a very important role in the entire life of a particular cell. Variety of metabolic pathways, the organization of the genome or the various structures is also being governed by cell signaling. Whenever there is certain changes in the environment, it can be a change in the temperature, it can be change in the availability of the nutrient, it might be the accumulation of the toxic substances. Whatever environmental clues are there, these are being accepted by the cell, and cell will respond accordingly via the cell signaling pathways only. It might involve entire cascade, and this entire cascade is rarely unidirectional. It is multidirectional. It diversifies at certain stages. It converges at certain points. One signaling cascade molecule of one particular pathway may crosstalk with another pathway. These are the important features of the cell signaling cascades. One of the most important signaling cascade receptor is GPCR, followed by RTKs, followed by JAK-STAT pathway. The JAK-STAT pathway is being used in the case of cytokine receptors. In a nutshell, we can say that whenever a cell needs to talk to another cell or needs to talk to oneself, it will do it by cell signaling processes only. So this is the conclusion of the entire cell signaling pathway, and I wish very best for the coming CSIR UGC NET examination, and I hope that this particular lecture on cell signaling will be helpful and more of the students can clear their NET exam through this particular lecture. Thank you.